Understanding head turns in dogs. Expanding beyond the forebrain, the study, The Value of a Head Turn in Neurolocalization, published in the Journal of Veterinary Internal Medicine, seeks to challenge the long-held assumption that a head turn in dogs is a primary indicator of forebrain disease. The research, a collaborative effort across multiple veterinary institutions, aimed to investigate whether the presence of a head turn, along with other postural deviations like head tilts and body turns, could effectively pinpoint the location of neurological lesions in dogs. This research is especially important because a head turn, or the more encompassing term pleurothodonus, has often been used interchangeably with other terms like torticollis and scoliosis, leading to potential confusion in diagnosis. Methodology and study population. The study involved 49 dogs exhibiting a head turn upon initial presentation, with photographic or video evidence. Each dog underwent a comprehensive neurological examination conducted by a board-certified neurologist or neurologist in training. Advanced imaging, using either magnetic resonance imaging (MRI) or computed tomography (CT), was performed to precisely locate lesions and determine the affected anatomical structures. To ensure clear results, dogs with multifocal lesions or lesions spanning multiple brain regions were excluded from the study. Based on the imaging results, the 49 dogs were categorized into 4 groups based on the location of their lesion: forebrain, 24 dogs, brainstem, 9 dogs, cerebellum, 9 dogs, cervical spinal cord, 7 dogs. The researchers also documented the dog's signalement, suspected or confirmed diagnosis, and the specific types of lesions present. This included lesions like intra-axial solitary masses, ischemic infarcts, extra-axial solitary masses, non-traumatic hemorrhage, atlantoaxial subluxation, syringahydromyelia, acute hydrated non-compressive nucleus pulposus extrusion, suspected seizure-induced change, intervertebral disc protrusion, and traumatic spinal cord injury. Challenging the forebrain paradigm. While the forebrain emerged as the most frequent location for lesions associated with head turns, a significant number of dogs exhibited lesions in other areas of the central nervous system. This crucial finding challenges the traditional notion that a head turn is solely indicative of forebrain dysfunction. Head turns in forebrain disease. In dogs with forebrain disease, an ipsilateral head turn, meaning the head turn towards the side of the lesion, was observed in almost all cases, 23 out of 24. The single exception was a dog with a suspected seizure-induced change in the piriform lobe, which displayed a contralateral head turned away from the lesion. The researchers suggest that this unusual presentation might be due to stimulation of the piriform lobe and amygdala, rather than a loss of function, as is typically seen in other cases of forebrain lesions. This theory is supported by previous experimental studies in cats, where stimulation of the amygdaloid nuclear complex resulted in a contralateral head turn. Within the forebrain, lesions were found in various areas, including the telencephalon, diencephalon, frontal cortex, temporal cortex, parietal cortex, occipital cortex, piriform lobe, and basal nuclei. The study authors propose several hypotheses to explain the occurrence of head turns in association with lesions in these areas: disruption of sensory processing, lesions affecting the frontal, parietal, and temporal cortices, which are responsible for sensory processing, could lead to a loss of normal suppressive input and result in co-contraction of muscles, potentially causing a head turn. Basal nuclei dysfunction. The basal nuclei, particularly the caudate nuclei, may play a role in abnormal head turning. Studies in humans have linked serotonergic depletion in the caudate nuclei and striatum to ipsilateral head turning. Thalamic involvement. Head tilts observed in some dogs with forebrain lesions, particularly those involving the thalamus, could be attributed to dysfunction of the mediadorsal nucleus of the thalamus. This nucleus has connections to the interstitial nucleus of Caudal incorporated in the cerebellum, both of which play roles in head posture control. Parido-insular cortex dysfunction. In humans and macaques, the parido-insular cortex integrates sensory vestibular input from the head, neck, ears, and vision. Lesions in this area could disrupt the sense of verticality, leading to alterations in head posture. Head turns in brainstem disease. In dogs with brainstem disease, head turns were always accompanied by either a head tilt, a body turn, or both. This underscores the brainstem's intricate involvement in coordinating head and body movements. The researchers suggest that lesions in specific brainstem areas could be responsible for the observed postural abnormalities. Interstitial nucleus of Caudal. Lesions affecting the interstitial nucleus of Caudal incorporated located in the midbrain, or the descending interstitiospinal fibers within the medial longitudinal fasciculus, could disrupt the excitatory synaptic connections to ipsilateral cervical muscles. These muscles, including the biventor, splenius, and sternocleidomastoid muscles, are essential for head movement. Substantia nigra reticulata, although not observed in the study. Previous research in cats has shown that ventrolateral lesions of the midbrain, specifically affecting the substantia nigra reticulata, can lead to a contralateral head turn. Dorsal raphe nucleus. The dorsal raphe nucleus is crucial for serotonergic innervation of the substantia nigra and rostral cortex. Disruption of this pathway, as demonstrated in studies on rats, can result in an ipsilateral head turn. Reticular formation. The reticular formation, which plays a vital role in regulating muscle tone, could be involved in the body turn observed in some dogs with brainstem lesions. Head turns in cerebellar disease. In dogs with cerebellar disease, lesions were most commonly found in the rostral vermis, particularly the culmen, lobulus, and declive. These dogs typically presented with an ipsilateral head turn and a contralateral head tilt. The researchers point out that while previous studies have described various head posture abnormalities in dogs with rostral cerebellar infarctions, including ipsilateral or contralateral head tilts and torticollis, the concurrent presentation of a head turn and tilt is a novel finding. The rostral vermis of the cerebellum is responsible for coordinating gait and postural adjustments of the head, trunk, and legs. Lesions in this area could therefore disrupt these functions and lead to the observed head turn and tilt. In dogs with lesions in the cerebellar hemispheres, head turns were observed with either a contralateral or ipsilateral head tilt. The researchers suggest that these findings could be related to the vestigial nucleus, located in the cerebellar hemispheres. This nucleus integrates spatial information from the head and body to regulate posture and also plays a role in the vestibular system, which is essential for maintaining balance. Head turns in cervical spinal cord disease. In dogs with cervical spinal cord disease, head turns were never observed in isolation. Instead, they were always accompanied by a body turn, and often a head tilt as well. The authors suggest that pain and altered muscle tone, resulting from the spinal cord lesion, are the primary drivers of these postural changes. Lesions affecting different parts of the cervical spinal cord can disrupt nerve signals to the muscles responsible for head and neck movement. Ventral horn and white matter damage. Damage to the ventral horn and white matter can cause innervation and atrophy of the ipsilateral paraspinal muscles, leading to an imbalance in muscle tone and a deviation of the head and neck away from the lesion. Dorsal horn damage. Damage to the dorsal horn can interrupt sensory proprioceptive fibers that relay information from the joints, muscles, and ligaments. This disruption can lead to unopposed contractions of the contralateral spinal muscles, causing the head and neck to deviate away from the lesion. Pain originating from the cervical spinal cord lesion can also restrict the dog's ability to turn its head towards the affected side, further contributing to the head turn. The study authors observed that head tilts were present only in dogs with cranial cervical lesions, specifically those affecting spinal cord segments C1, C4. This finding is significant because the central cervical nucleus, located within these segments, receives input from neck muscle spindle afferents and projects to the contralateral cerebellum and vestibular nuclei, all of which play roles in neck rotation. Disruption of this nucleus could therefore contribute to head tilts. The study also included 2 dogs with atlantoaxial subluxation, a condition that can cause damage to the greater occipital nerve, a branch of the C2 nerve, or the C2 nerve root. These nerves innervate the sternocleidomastoid and trapezius muscles, which are crucial for rotational control of the head and neck. Damage to these nerves can therefore lead to both a head turn and tilt. Implications for veterinary practice. The study's findings carry significant implications for veterinary neurologists and general practitioners. Head turn is not pathognomonic for forebrain disease. This research definitively demonstrates that a head turn is not exclusive to forebrain disease and can originate from various parts of their central nervous system. Thorough neurological examination is essential. A comprehensive neurological examination, coupled with advanced imaging techniques like MRI or CT, is crucial for accurately localizing the neurological lesion responsible for a head turn. Consider all potential causes. Veterinarians should approach cases involving head turns with a broader perspective, considering the possibility of lesions beyond the forebrain. This includes evaluating the brainstem, cerebellum, and cervical spinal cord as potential sources of the problem. Refined terminology. The study highlights the need for more precise terminology when describing postural abnormalities in dogs. Clearer definitions based on the specific combination of head turn, head tilt, and body turn would improve communication and understanding among veterinary professionals. Conclusion. This study challenges the traditional view of head turns in dogs and emphasizes the importance of a thorough neurological examination and advanced imaging for accurate neurolocalization. By recognizing the diverse origins of head turns, veterinary professionals can enhance their diagnostic accuracy and provide more targeted and effective treatment plans for their canine patients.